So today we'll be speaking about the atria and the ventricles, the cardiac muscles, and how they are coupled with the excitation and contraction, and very interesting physiological concepts on absolute refractory period and the relative refractory period about what it is and how it works for the cardiac muscle, and also how it differs from skeletal muscle. I'll be telling you in a due bit. So let's start. First, we all know that the cardiac muscle is composed of the atria and the ventricles, right? Now, atria and ventricles, they both act as synexium. What I mean by synexium is that they both coordinate each myocyte of atria or the ventricles, they coordinate together. And for this togetherness and for this together coordination, it is very important that some of the functional anatomy we should look at of the atria and the ventricles cardiac muscle tissue. Now, here is a picture of the cardiac muscle tissue, and you can see that in comparison with the skeletal muscle, there is a similarity, which is these striations that you can see, right? Please appreciate the striations, which is also found in the skeletal muscle. What is the difference from the skeletal muscle is this intercalated disc. Can you appreciate the intercalated disc? Now, this intercalated disc, just let me write down here, intercalated disc. This is nothing but it is made up of the fusion of the cell membrane of this myocyte with the adjacent myocyte, right? It is just a cell membrane fusion. Why this had to occur? Because of this fusion, now this intercalated disc acts as a gap junction or a communicating junction. Now, because it acts as a gap junction, it easily allows the flow of all the ions from one myocyte into the another. Whenever an action potential strikes this myocyte, definitely it is going to allow easy transfer of ions from this to this myocyte to the adjacent, so that this adjacent myocyte also gets excited easily. Now, this is necessary to work in synexium. Now, as we know that the atria and ventricles are both a different synexium, how do you think that the atria transmits the action potential to the ventricles? Obviously, it's not directly, because atria and ventricle, between them there is the AV opening, atrioventricular opening, which has the AV valve, and it is surrounded by the fibrous tissue. And this fibrous tissue is not permeable in normal circumstances to the action potential traveling from the atria into the ventricles. So normally the route of transfer from the atria to the ventricles, the action potential travels by the AV bundle, the atrioventricle bundle of fibers, right? So this was about the functional anatomy, which was important in the cardiac muscle and how it differs from the skeletal muscle. So now let's talk about the cardiac action potential. We are talking about the non-pacemaker action potential, which is totally different from the pacemaker action potential. Now, speaking about this action potential, you should appreciate the difference between the skeletal muscle action potential and the cardiac muscle action potential. Now, the skeletal muscle action potential has a similar depolarization spike. It has the repolarization arm, but what it does not have is that it is lacking this plateau phase. And this plateau phase, because of the lack of this plateau phase, we can say that the cardiac action potential has a much longer duration of the sustained action potential than the skeletal muscle action potential, right? Now let's talk about the ions in each phase. You should know that the depolarization spike is because of the predominance of the opening of fast sodium channels. Now we all know that the fast sodium channels also opens in the skeletal muscles, right? But what is the difference between the skeletal muscle and the cardiac muscle in the depolarization phase is that there is also predominance of the calcium ions which diffuses inside the myocytes, whereas this does not happen in the skeletal muscle. And this occurs with the help of slow calcium channels, right? So now you know the depolarization arm are predominant with these two ions. After the depolarization phase, we know that the sodium channel, it closes. Sodium channels close. Now, after the closing of sodium channel, appreciate this phase here. This phase is the early repolarization phase. It takes place because of the dominance of potassium ions. Now, that means the potassium ion channels have opened and the potassium ions have started to diffuse outward of the myocyte. And this potassium ions gradient is so significant that although the calcium ion channel is opened, calcium is coming inside, but and with a +2 charge that too, but still the potassium ions gradient is so significant that so much of potassium with a high flow rate, with a high flow velocity, it is diffusing outwards, which is why there is this drop in the potential and it is trying to bring the potential to the nearer to the membrane potential. We know that the potassium ion channels have opened and it tries to bring the membrane brings the potential towards the membrane potential and has started repolarization. But we should not forget the fact that the calcium channels are still open now, right? So since the calcium channels are already open, this is going to balance out the effect of repolarization, which the potassium channel is trying to bring. So in this plateau phase is what I was trying to explain, that there is an potassium channel which is going to diffuse outward, plus there is going to be calcium channels which are going to diffuse inwards, right? Which are going to diffuse inwards. And because of this inward diffusion of calcium ions, what is happening, it is trying to balance out the effect of the potassium ions going outwards. That is why this balance in potential, and therefore this is leading to the plateau phase and sustained action potential. Now, after this, what happens is calcium channels are going to close and only potassium channels are going to predominate in the repolarization phase of the action potential of the cardiac myocyte. So this is how the skeletal muscle uh, action potential differs from the cardiac muscle action potential. Now, after this, let's talk about very interesting absolute and relative refractory periods. We all know what is a refractory period. A refractory period is a period where once an action potential is generated, for a second action potential to be generated, there has to be a particular interval. That means there has to be a particular duration of time, which is for a cardiac myocyte zero point 0.2 to 0.3 seconds. And for this zero point 0.2 to zero point 0.3 seconds, once when an action potential is generated, another action potential cannot be generated for this particular interval. It has to wait for zero point 0.2 to zero point 0.3 seconds, and then another action potential can be generated. Now, this is known as the refractory period, and this is the absolute refractory period duration of the cardiac myocyte, right? So look at this, focus at this picture here. I'll just zoom it. Okay. So if you look at this picture here, you can see that the absolute refractory period is this one, right? This is the duration of 0.2 to zero point 0.3 seconds in the cardiac muscles is this absolute refractory period. And then you can also note this particular, interval or duration, which is around 0.05 seconds. Now, this 0.05 seconds is what is known as the relative refractory period. Now, what is the relative refractory period? This is differing from the absolute refractory period in the sense that this is also a refractory period. So here again, a second action potential cannot be generated, right? But theoretically, it is said that in the relative refractory period, an action potential can be generated with a much greater threshold, with a much greater force required. That means it would be difficult to initiate an action potential in the relative refractory period, but it is possible, right? So this is why it is known as a relative refractory period. Okay, so and you can appreciate in the second picture, second graph here itself, that the relative refractory period, this 0.05 second, you can definitely initiate an action, second action potential after the first one, and that is what leads to an early premature contraction, right? So it was possible to generate, but it is difficult to generate. That means a much greater force is required to generate the action potential in the relative refractory period. In the absolute refractory period, you can never generate an action potential, a second action potential, but in the relative refractory period, you can definitely generate one, but it requires a much greater force. And that is what leads to an early premature contraction. In this 0.05 seconds, there has been an action potential which is generated. Right. So after talking about this, Let's focus on something which is excitation contraction coupling. Okay, let me set the picture for you and then we can start. So here you go. This is the excitation contraction coupling I was talking about. So we all know that once the action potential reaches the cardiac myocytes, right? With the help of gap junction, it is going to, in the first picture, we already studied that with the help of intercalated disc or the gap junction, it's going to spread to the other myocytes. But what happens actually here is that within one myocyte, what is happening? We should look at that. So within one myocyte, we can see that this is the sarcolemma or the muscle's cell membrane is known as the sarcolemma. This is the sarcoplasmic reticulum, which is the muscle's endoplasmic reticulum, right? And within this sarcolemma, there are something which is known as T tubules, okay? These are very important structures. Now, these T tubules are the ones which are having some specialized ion channels, right? For example, in the skeletal muscle as well as in the cardiac muscle cells, there is the presence of this dihydropyridine receptor, right? Now, this dihydropyridine receptor, for example, it is going to be excited when the sarcolemma or the muscle cell membrane is going to be excited by the action potential. And when this DHP receptor is excited, this is going to activate the calcium release channels, also known as the ryanodine receptor channels, in the terminal cisterna of the sarcoplasmic reticulum, right? And this sarcoplasmic reticulum, it is a storehouse of calcium. There are lots of calcium ions inside the sarcoplasmic reticulum. And because of the calcium release channel being activated, this calcium is going to go outside into the sarcoplasm, which is the cytoplasm of the muscles. And therefore, it is going to lead to the mechanism of contraction. Right. This is what is happening in a skeletal muscle, which is also taking place in a cardiac muscle cell. But what is significant is that in a cardiac muscle cell, apart from this mechanism, there is also the presence of calcium channels, which is present in the extracellular fluid of the T tubules. Now, these calcium channels you can see in the T tubules, you can appreciate them. So in the cardiac muscles, additionally, even the calcium ions itself from the ECF goes inside into the sarcoplasm, and they lead to the activation of this calcium release channels or the ryanodine receptor channels, right? So as for now, to summarize, we have studied these two particular mechanisms, one which is present in both skeletal as well as the cardiac muscle, which is via the route of the action potential traveling in the T tubules, and with the help of dihydropyridine receptor, it is going to activate the calcium release channel. And the other mechanism, which is only specific to the cardiac muscles, which is that the calcium in the extracellular fluid itself is going to diffuse inside the sarcoplasm with the help of this calcium ion channels, which are present specifically in the cardiac muscles T tubules. And it is also going to activate the calcium release channels or the ryanodine receptor channels, with the help of which sarcoplasmic reticulum is going to release its storehouse of calcium into the sarcoplasm, and it will lead to the contraction, right? So this much here is clear. And it is very important because if we see particularly the anatomy, then you should know that the cardiac muscle sarcoplasmic reticulum, it is a storehouse of calcium, but it is the calcium ions are fewer in the sarcoplasmic reticulum of the cardiac myocyte than the sarcoplasmic reticulum of the skeletal muscles. That is why it needs the secondary mechanism of the calcium in the ECF itself being going to be diffused inside the sarcoplasm. So it needs additional calcium ions from the extracellular fluid itself. Now, for the calcium ions to be present in the extracellular fluid, specifically near the T tubules, the T tubules here have some mucopolysaccharides. It has these mucopolysaccharides which are negatively charged, and this negatively charged mucopolysaccharides allow the calcium in the extracellular fluid to remain here, and their availability increases so that easily whenever an action potential comes, these calcium ion channels are going to be activated, and all the calcium which is available here with the help of mucopolysaccharides, this calcium will diffuse inside. Okay, so this is how the calcium comes inside and the contraction occurs in a cardiac muscle. Now, what happens when has to be relaxation? For relaxation to occur, this calcium which is present in the sarcoplasm, these calciums, they are going to either go inside the sarcoplasmic reticulum. This happens in the skeletal muscle. Now, since the calcium during contraction had come from the extracellular fluid, it has to go back to the extracellular fluid, and this occurs with the help of the calcium sodium exchanger as well as with the help of calcium ATPase pump. Now, we also know that the sodium, with the help of fast sodium channels during depolarization, when they had come inside, with the help of sodium potassium ATPase pump, also the sodium will be pumped outside and the potassium will be pumped inside, right? So this is happening and the calcium availability is going to decrease, and due to which there is relaxation phase which is going to take place. So this is clear, right? Now, how does the contraction actually occurs? Now, this is similar to the mechanism of the skeletal muscle. There's no differentiation, and let's talk about this contraction mechanism which is taking place in the skeletal muscle as well as in the cardiac muscle cells. Now, what is happening? We know that for the contraction to occur, the myocytes are made of special filaments. This is the myosin heavy chain filament, and this is the actin light chain filament. Now, we all know that the actin and myosin, they have to form a cross bridge. You can appreciate the cross bridge here. So this cross bridge has to be formed for the contraction to occur, right? So for this cross bridge formation, actin has some particular active sites. Actin filaments, you can see this purple beaded active actin filaments, they have some particular active sites present on them, right? Like this. Now, this is the active site to which the myosin filament or the myosin head is going to bind to this active site. But these active sites are covered with the tropomyosin molecule. So these blue chains here are the blanket to the active sites. That means they are covering the active sites in the form of a blanket, these tropomyosin, and with the help of which these active sites are only exposed when there has to be a contraction, right? Until then, they are covered. Now, when the tropomyosin covers this active sites, obviously there has to be something which has to pull the blanket or pull the tropomyosin molecule, and there is where the role of troponin comes in. Now, there are three types of troponin. There is troponin I, there is troponin C, and then there is troponin T. Now, the troponin C was going to bind to the calcium ions, and we know that the calcium has to be present for the contraction. That is what we just saw previously. So when the calcium comes and bind to the troponin C, the troponin I and T. Troponin I is bound to the actin filament and troponin T is bound to the tropomyosin filament. There is such configurational changes which is leading to the removal of the blanket which is spread out by the tropomyosin molecule, and the actin's active site are being exposed, and now also the ATP which is available here is going to break down, and the myosin head is going to bind to the actin's active site, right? Now, as we can see that the myosin heads, they look like this, okay? So just appreciate my diagram here. For example, this is the myosin head, okay? And here goes the actin filament. And now this actin filament, this myosin head binds to the active site on the actin filament, right? Once it binds, it is going to pull the actin filament to this side. To this side in relation if the M line is here. That means from both sides, the myosin filament is going to pull it towards the M line, towards the center of the sarcomere, right? It will try to pull it toward the center of the sarcomere. Now, this is going to be done by each of the myosin heads. All the myosin heads are going to pull all the actin filaments which they are binding independently towards the center. Now, once they start pulling, now for example, here again, one more myosin head is there, it is going to also pull it towards the center of the sarcomere. Now, once they start pulling, the direction of the pull becomes the side of the of the M line and the side of the M line, right? If this is the M line, for instance. Now, once this happens, what happens is that the head tilt is going to occur. That means this myosin filament, once bound here, and after pulling it here to this side, this myosin head is going to be tilted in this direction, from this direction to this direction. Now, then it is going to leave this attachment of the active site, of the initial active site, and after leaving, it is again going back to its initial position. For example, was the head. It came in this direction after the head tilt, and again it went back to its original position. Now, once it goes back to the original position, it is going to bind to another active site. Again, it will lead to the pull. Again, it will go back. So, with the help of this, each of the myosin head are trying to pull the actin towards the center of the, and this is how the contraction or the sliding filament theory, this is what it how it works, right? And this mechanism of pulling the head towards the center is known as the power stroke, right? This is the power stroke mechanism. This is the term power stroke, which is known as. So this was all about the topic today, which I was going to speak about. So let's meet in another video lecture where I'll be speaking about skeletal muscles and how the cardiac cycle works, and all these will be taken to the next lecture. Thank you.